the role of immune responses and inflammation in neurodegeneration. So this is just to remind me, to remind you that inflammation is extremely complex, and we have this term which really doesn't mean much. Inflammation consists of a natural immune response that primarily tries to solve a problem that occurs in our bodies, and part of this response is beneficial, part of it is detrimental, and frequently we don't know exactly what we should target. But just with respect to the brain, in an injury situation, we have various stages of injured neurons that release signals that activate the local immune cells. And really the microglial cells are classical macrophages that are capable of producing all the cytokines, chemokines, complement proteins that are produced in systemic inflammatory reactions. Similarly, the astrocytes are very capable immune cells, and they can produce most of these factors. And I just threw a few of these molecules here, receptors such as complement receptors, CD14, Fc receptors that are able to recognize dead cells or sometimes protein deposits extracellularly but also intracellularly. And all of these play maybe a more important role in classical neurodegenerative diseases, but I think also in other forms of tissue injury. We have all kinds of proteins that trigger TLR receptors, for example, on immune cells. Now, it's much rarer, so this is the typical innate immune response, as we call it, but occasionally the adaptive immune response is recruited as well, and the question came up previously, when does that happen? For example, in neurodegenerative diseases, we know in ALS or in Parkinson's disease mouse models, T lymphocytes have either beneficial or detrimental roles. So even in classical neurodegenerative diseases, there seems to be a role for these immune cells, and we're still trying to understand what they're actually doing. But what we try to do here, and what I was invited to discuss, is how can we get an understanding of these responses in a living organism, specifically in a disease model. So what I plan to discuss with you today is the use of non-invasive molecular imaging tools for neuroinflammation, and in the first part talk about imaging of a specific signaling pathway, of a cytokine signaling pathway that is involved in neuroinflammation, and give you examples of monitoring this immune signaling, monitoring neuronal injury or neuroinflammation, if you will, the use of these models for studying therapeutic efficacy or for localization of the injurious response or inflammatory response. And then also talk about one model that we used in particular that monitors astrocyte activation. And then I have just one slide of models that people have developed and we are developing to monitor microglial cell activation. So first, bioluminescence imaging to study a specific signaling pathway, and we've heard a lot about interleukin 1 and 6 and TNF-alpha, but I'm studying the good guy here, or at least we think so, transforming growth factor beta, and like the pro-inflammatory cytokines, this classical anti-inflammatory cytokine is produced in every injury response you have in the body. It's produced by all cells. In the CNS, every cell expresses receptors for TGF-beta, and so can potentially respond to it. Our interest in trying to make a reporter mouse for this pathway came from observations that if you overproduce TGF-beta, you get a number of pathological changes in the brain. We found, for example, that astrocyte overproduction of TGF-beta leads to amyloid deposition in the vasculature. It leads to a tremendous activation of the astrocytes around the blood vessels, but it's also neuroprotective, so it has a lot of different effects, and this is just one way of illustrating how the immune response is frequently Janus phased and can do different things. So in order to study this and find out where does the signal come from and how it develops over the course of the disease, we developed a mouse model that reports activation of this pathway. So just very briefly, if TGF-beta binds to its receptors, it leads to phosphorylation of so-called SMAD proteins that translocate to the nucleus where they bind to the SMAD binding element, which is present in a few hundred genes. And that regulates the expression of these genes. So if we take multiple elements of this binding site, DNA binding site, and fuse it with a luciferase gene, we have a reporter that now, if we activate TGF-beta signaling, we not only produce normal genes, but we also produce luciferase. So wherever in the body TGF-beta signaling gets activated, these cells will produce luciferase. And why do we put luciferase in there? First of all, luciferase is an extremely sensitive reporter that you can homogenize a tissue, for example, or a cell, and you add the substrate, which is called luciferin, and you can then measure the enzyme activity per milligram tissue. This is what we did here in 2 independent lines of these transgenic mice. And what we noticed when we looked at the relative activity, so this is just a normal mouse that expresses this reporter, we found that the brain by far has the highest basal activity for the TGF-beta signaling pathway. Now this was novel, and TGF-beta had not really been studied much in the CNS. So this was very reassuring for us that this is an interesting pathway to study. The second most prominent organ was the intestine, and I have a couple of slides that talk to that later. But this is an example of just looking at the tissue activity for this enzyme to find out where is this pathway activated. Now, obviously, you can do that in disease as well. But why we use luciferase is really for the reason that it glows. If you give the specific substrate, you produce bioluminescence. And here, this is a firefly, a picture taken with a flash. So this is how the fly looks. And here, if you turn off the flash, you see this bioluminescence that is generated from this organ here. And the reaction is quite simple. Luciferin together with oxygen, ATP, and luciferase produces oxyluciferin and light. And so you can see these photons, and of course you can count them. So this is really the idea of the bioluminescent mice. And this was not developed, but it was certainly the approach was pioneered by Chris Contact at Stanford. And that's just about when I moved to Stanford. So I asked him if he could help us image our mice and see whether we get a signal in the CNS. So this is just a dark box with a very sensitive camera. You put the mice in there. You inject luciferin, and so luciferin distributes throughout the mouse, and wherever luciferase is present and where living cells are present that have oxygen, you produce photons which you can record, and you get signals like this. So this is the baseline signal from our TGF-beta reporter mouse, and you see, as consistent with our tissue analysis, the major signal comes from the brain. So we use then an injury model that activates excitatory channels, glutamate receptors. We inject kainic acid, and that causes synaptic degeneration, apoptosis, cell loss, and also prominent neuroinflammation. And this is a model that we can use to try to study the activation of this pathway. And I show you here as an example how you can use these mice as a tool to image neuroinflammation. So here is a non-transgenic mouse. You get no photons produced. With saline, as I showed you before, there is a weak signal that we detect over the brain. But then if we injure the mouse, this is 24 hours after the injury, we get a very nice signal consistent with activation of the TGF-beta signaling pathway in these mice, specifically in the brain. We can take out the brain, slice it up, and put it under the camera, and you can see here again, this is the saline image, and this is the kainate-injured slice, and you see very nicely the majority of the signal comes actually from the hippocampus. This is the main target of kainic acid. That's where you get the largest neurodegeneration with this drug. So this suggests that what we're imaging here is truly a molecular signal that corresponds to activation of this pathway. So obviously what we would like to do is quantify that signal and follow it over time. So here is again how this signal, as I mentioned to you already, it targets mostly the hippocampus. Here you see a control hippocampus, the CA1, CA2, CA3 region, and the dentate gyrus in a Nissl stain. If you give kainic acid to the mice, you get a prominent degeneration of CA1 and also CA3. This is after 5 days, so you get a striking loss of the Nissl stain in these neurons, and they eventually die. Now, the real advantage of the bioluminescence imaging is that you can follow the animals over time. And you can, especially in a model, and it seems that in the pain world this is the case as well, there's a lot of variability. And this is certainly the case for the kainate model. So I show you just 2 mice here that have various degrees of injury. This one has almost all neurons lost. This one has still a substantial number of neurons left, and this is the uninjured ones. So there's different degrees of activation, and the TGF-beta reporter here, again, is a surrogate of this injury, a surrogate marker of this injury. But can it help us to quantify the relative damage that occurs in these animals? That's the question. So one of the prominent features of this model that I mentioned to you is neuroinflammation, microglial cell activation. We use here a marker that is called CD68. It's expressed in the macrophage lineage. It's a lysosomal protein, and it's very prominently activated when microglia get activated. You can see here in a normal mouse, there is almost no staining for CD68. And then here I showed you 3 extreme examples of mice that have various degrees of degeneration or injury after kainate. And you can see various degrees in the cortex of microglial activation. You see these very round amoeboid microglial cells, and also here in the CA1 region where we get this very prominent damage. So if bioluminescence is supposed to be useful, ideally the signal that we measure in the mouse before we sacrifice it, before we make these slices, correlates with the pathology that we see later on. And this seems to be indeed the case. We get a fairly good correlation of 0.8 here between CD68 immunoreactivity measured at day 5 and the bioluminescence that was measured 24 hours after inducing the injury. So the bioluminescence gives us an indication of the relative degree of damage that each mouse experienced. And again, certain mice have very little damage. They have less CD68 expression, and others have a lot of damage. And that seems to correlate with activation of our reporter gene, TGF-beta. So can we use this to monitor therapeutic efficacy? It seems to be the case because in this model, this is still the kainate model. I mentioned to you the injury is a glutamate receptor agonist, and MK801 is, of course, a very good inhibitor of this receptor. So if we give MK801 to animals, we can dramatically reduce the bioluminescent signal. And we can then later confirm pathologically that these mice are also protected. They have very little damage compared to the non-protected animals that just received kainate and PBS. So you could basically test, with this model, you could test other drugs that would prevent this injury, and we have indeed done that. We're studying macrophage colony stimulating, for example, factor, which we find is strongly neuroprotective, and we actually use this model to test when we can give the drug. Can we give it after the injury? How long after the injury can we give it? So this is really a model also to test to screen for drugs because it's relatively fast. You can image 5 mice at a time, and you have a relatively high throughput. Now, you can also use bioluminescence imaging to localize where the injury is if you don't know, such as in a syndrome where there is some disease going on, but you don't know exactly where it is. We use here the LPS model. This is still the TGF-beta reporter mice. If you inject LPS, you get inflammation basically in any tissue, but the degree of inflammation is different. And here we measured bioluminescence in various tissues. So we actually took the tissues out right after. So we inject LPS, then we inject luciferin 24 hours later, and take out the tissues and measure the relative bioluminescence activity in these tissues. And you can see again the brain has the highest signal in response to LPS. The intestine shows a 600-fold increase over baseline. So a tremendous activation of the TGF-beta signaling pathway in the intestine. And you can do this also with non-invasive imaging. Here we flip the mice on the back, and you can see that indeed in the intestine you have a very strong signal that comes from the activation of the reporter there. So this is consistent with the tissue analysis. If we do a time course, we can look here activation of at the progression of this signal here now over time. This is 5 hours after LPS injection, but we can do a time course, and what you see is a remarkable fast increase at 5 hours peak, and then it goes down 12, 24, 48 hours. It's back to baseline. So this can help you to look in a model where you're not sure exactly when you have the highest degree of injury, when is your reporter most activated. This is a relatively easy way to do non-invasive. You can image the same animal over time, multiple times, to get these type of curves. What was interesting here is that this is such a transient signal, whereas in the brain, the signal seems to stay up. So here we get a 3 to 4-fold increase over baseline. But when we look over time, there seemed to be a chronic persistence of this signal in the brain. And again, that might tell you something about the type of response, injury response you have. And it's very simple to image this. So let me then switch to the other model. So this was molecular imaging of one specific signaling pathway, and you can literally take any gene of interest, you take the promoter and you fuse it with luciferase, and you get a pathway-specific reporter mouse. And this has been done. I'll show you in the end a slide with a few other examples for a number of different signaling pathways. But you could also take your gene of interest, such as it has been done for astrocytes using the GFAP promoter. As was noted earlier, GFAP is extremely injury responsive. It's a very good indicator of any form of brain injury. Astrocytes become activated, and as part of that activation, they induce transcription of GFAP. You can link any gene behind this that you want to produce in astrocytes. And here, this is a mouse that was made at Synogen. So this is a company that was started by Chris Contact. It was now purchased by Caliper. You can buy these mice at a tremendous costs with a contract that is quite restrictive, unfortunately. But I think you have to try to get into a collaboration that may be easier. So what we did here is we got these mice that were generated with a GFAP promoter fused to luciferase, and we used them for similar types of imaging, and now we just image activation of astrocytes in general. Similar to before, what I said before, you inject luciferin, you image them in these. And here I show you an example of an autoimmune disease. This is a mouse model that is used to study multiple sclerosis. It's called EAE. And what you do is you immunize mice with myelin, different myelin proteins. There are different models. We use MOG in black 6 mice. Together with adjuvants, you immunize the mice. And after a few days, you get activation of this GFAP reporter, again as a surrogate marker of the type of injury that occurs in this mouse. We've done the same imaging with our SBE-luciferase, TGF-beta pathway-specific reporter, and we get very similar results because most of these reporters are injury responsive, so this is not surprising. But this is just, if you look at the pathology in this mouse model, so at baseline there is very little expression of GFAP. Now we look at protein, little expression of CD68, as I showed you before, as a marker for microglia, no T cells or barely any T cells in these brains. And then here, 3, 7, 10, 14 days after immunization, our model usually peaks with a clinical disease at day 14. And you see activation of astrocytes and microglia actually preceding the infiltration of T cells. And what's interesting is that we can monitor this with the bioluminescent imaging approach. This is shown here. We can actually show the clinical score here is shown in green. And the clinical score really correlates very nicely with the infiltration of T cells. So you see the first clinical signs of paralysis when you detect T cells in the spinal cord and the brainstem and the cerebellum. But usually you don't see any symptoms at this earlier stage, at least not the crude symptoms that are used, the paralysis. We actually see sleep disturbances in these mice at earlier time points, which is kind of interesting. But when we use the bioluminescence reporter here, the activation of GFAP, you can see consistent with the pathology, astrocytes get activated very early. You see the first signals in the brain at day 7, possibly even earlier, and then they sort of flatten out. This is the same mouse imaged over time. So again, gives you an example that you can follow an individual. You can do an intervention. You can see whether this makes it worse or better. So you can reduce the number of animals in your study, but you can also study disease variability where you may not get significant results. Here you can always correlate it back to your baseline expression, and that's really very nice. And so lastly, other mouse models, so mouse models to monitor microglia would of course be very useful. We are trying to make a CD68 luciferase mouse. This is one of the first mice we have. We have to test it first. If it works, it has a low signal over the brain. But this promoter is not specific to microglia. It's also expressed in the macrophages in the periphery. So you would expect signals everywhere. But maybe the microglia in the brain have slightly higher expression of this gene. We want to injure these mice and see whether we can use them as a specific reporter for microglial activation, because we think frequently microglia are more sensitive to certain types of injury than astrocytes even. This is an example from Dr. Chris in Quebec. And what she did is she used the TLR2 promoter. So this is a toll-like receptor that is an innate immune receptor, very responsive to activation of activated in macrophages, fuse it to luciferase, and you can see here a very low signal over the brain in untreated animals, and then 6 hours and 24 hours after injection of LPS, there's a very nice induction of the reporter gene. So this is another example of trying to monitor activation of, in this case, cerebral ischemia. A few other models that have been generated. CEBP luciferase is a very recent paper. Serum amyloid A luciferase, INOS luciferase, and NF-kappa B luciferase. So these are mostly pathway-specific but they all fall sort of in the category of neuroinflammation. Of course, this approach has been used to study a lot of different genes in a lot of different disease models, or just in normal physiology. But I just list here a few examples of mice that could be used to study neuroinflammation. And they would certainly be, some of them you would expect would be activated in the models of neuropathic pain or chronic pain syndrome that you're studying. So to summarize and conclude, I think bioluminescence imaging in mice can be used to report activation of specific genes, specific to signaling pathways or to cell types. It can be used to quantify injury and inflammation, both temporally and spatially. You can use it to localize sites of injury, and it can be used to study drug efficacy and pharmacology. And as such, I think it has a lot of potentials. Unfortunately, it's, I think, rather unrealistic to use this approach in humans unless you make an engineered reporter, a viral reporter or something like that, where you would want to track, for example, macrophages for therapeutic purposes. But I think it's mostly really confined to animal models. I'd like to acknowledge people who did the work. Chen Luo in the lab did most of these animal studies together with a grad student, Ching and Kira. And then collaborators, I mentioned Chris Contact, but also Sam Gambier is now chair of radiology at Stanford, and Larry Steinman with the EAE model together with Peggy, and then outside collaborators and funding sources. Thank you.
It's known to be messy and patchy, just like in MS. And what you're showing was almost overall activation within the spinal cord and the brain that comes all at once, which is a little bit counterintuitive also with the motor deficit, which starts with the tail progressively to the hind limbs and then moves up. Any comments on that?
Yeah, so I think you're right. Most people thought that the spinal cord is affected first and then the brain, but it has really not been studied well, I think, and it has not been studied with these types, with the perspective of the CNS. I think traditionally EAE was a model of immunologists, and there's very few that study the neurodegenerative aspects. A few neuroscientists who study those, and they frequently don't talk to each other. I think it's getting better, but the idea that maybe the microglial and astroglial response are actually earlier in this model, precede a T-cell response, I think that's a good possibility. And obviously this is a very artificial model. Whether that relates to MS is not clear, because we use a very strong innate immune trigger with the CFA that initiates this innate immune response.
I think as we appreciate how often there's microglial activation, we're all impressed how little perturbation activates the microglia. And I was shocked to see that a saline injection led to upregulation of the TGF-beta. Was that also true of the GFAP astrocytes? And could it be just the injection of the MOG caused, as the saline controlled it? It was remarkable. A saline injection activated the TGF-beta pathway, yes?
Well, so this was not, the saline control is basically the baseline that we see in this model. So whether we inject saline or nothing, we see a baseline.
But the saline was above your don't inject anything control.
In the EAE or?
No, in the TGF-beta. When you first showed us the TGF-beta data, the saline was above no injection at all. And then you didn't tell us in the GFAP model whether saline...
In this one?
Yes.
16.
See?
No, slide 16.
That's nothing, and then saline.
Slide 16. Okay, sorry, I'm a bit slow.
There you are again, yeah.
Yes, so this is a non-transgenic mouse. Sorry, it's maybe a bit confusing. This is a non-transgenic mouse injected with kainate. So a non-transgenic mouse never has any signal. That's the beauty of bioluminescence imaging. There is zero background. And then this is our transgenic mouse, the luciferase transgenic, and that has a baseline signal whether we inject saline or not. Sorry, that's confusing. But I just put here this non-transgenic mouse to show that there is no background with this tool, with this method.
Richard, brief response to Carl's comment that EAE is patchy. I think if you go through the literature, it's quite well described that it is involving the whole CNS more or less instantaneously. Now, I have two questions. One is, in your EAE models, did you look into the intestinal system? Did you see any changes as with LPS? That's one question. And the other question is, does the luciferase itself cause cell biological changes? There's something known for GFP, that it does alter cell reactivity.
Yeah, good questions. So we have not looked at the intestine, but it's a very interesting question. Obviously, the astrocytes, where we report from, or also the TGF-beta pathway, are very prominently activated. And the astrocytes are present in all the peripheral tissues as well. We have not looked at that, but we should probably have a look at it. It's not a strong signal, and the tool is not as sensitive as you would like it to be. Here we signal through the skull. And this is quite remarkable that we actually get a signal. People first thought it would not work in the brain. Your second question, I'm not aware of any studies that looked at specific side effects of luciferase, but I could imagine that if you had high levels of luciferase activity because you use oxygen, that this could have a detrimental effect. We give a single bolus of luciferin and image for maybe, you can image for up to an hour or so, and then the signal is gone. We have not noticed an acceleration of neurodegeneration, for example, when we compare non-transgenic animals that were given kainic acid, for example. But it's a good question.
Sorry, Nick?
If you want to do crossover studies or follow-up studies?
Yeah, so here we may have some protection from that, but it's a concern as well. We thought about this, especially if you express it within the CNS confines, and then if you have an injury like EAE, you could have, display to the immune system, to T cells and dendritic cells, and induce an immune response. It's a good point.
Tony, I'd like to ask one question I put in my intro this morning. Mm-hmm. The CRPS research community would really like to develop a rodent model for CRPS that matches human phenotypes more closely than the current models that are available. The new information that has emerged is that many CRPS patients have autoantibodies, and one of the autoantigens is the beta-2 adrenergic receptor. The autoantibody binds to the second extracellular loop domain. So do you think it's feasible to take that domain and immunize this mouse strain to create a similar situation that you've done with this model for the MS disease?
Yeah, I don't see why that wouldn't work. I mean, you can, if the homology with the endogenous protein is not too big, you should be able to induce an immune response. I guess Rick would be better able to speak to that. But these mice you can breed them to any background strain. It doesn't really matter if you could, cross them to a BALB/C that might be more immunogenic, or where you get more autoimmune disease and NOD background. That's absolutely possible. Yeah.